My name is Kirsten Hannam. I'm an agroecologist with Agriculture and Agri-Food Canada at the Summerland Research and Development Centre in the Okanagan Valley of British Columbia. I'm going to give a presentation today that I've called Growing Crops, Building Soil Carbon. I'll be presenting the results of a regional study I and my colleagues have recently conducted in grape, apple, and cherry cropping systems here in the Okanagan Valley. Before I start, I wanted to mention that this work is a product of a large collaborative relationship between Agriculture and Agri-Food Canada scientists in Summerland, BC, and in Harrow, Ontario, as well as research scientists at the University of British Columbia, Okanagan in Kelowna. I'm going to try and address three main questions today. First, why do we care about soil carbon? Next, how does soil carbon storage vary among apple orchards, cherry orchards, and wine grape vineyards here in the valley? And then, how could changes in management practices affect soil carbon storage in the future? Could we use management practices to increase or decrease soil carbon storage? So let's take a step back and look at the global carbon cycle to get a better understanding of why we even care about soil carbon in the first place. You can see from this figure that the ocean contains by far the largest amount of carbon, about 38,000 petagrams. But the next largest carbon pool is the soil. It contains about 1,500 petagrams, and that compares with about a little more than half the amount of carbon in the atmosphere. We know that fossil fuel burning has increased the amount of carbon in the atmosphere, but so have a lot of other practices, such as soil disturbance, which promotes the decomposition of soil organic carbon, that humus or organic matter that makes healthy soils really dark. It promotes the decomposition of that material and its release as carbon dioxide or CO2 into the atmosphere. So it accelerates the whole climate change phenomenon that we often associated with fossil fuels. Because the soil carbon pool is so large, small changes in the amount of carbon stored in the soil have big implications for the global carbon balance. And we see that a lot in the news. Changes in the amount of soil carbon caused by increasing temperatures, disturbance of the soil, promote the release of CO2 into the atmosphere. But because the soil carbon pool is so large, we can also use changes in soil carbon as a climate change mitigation tool. Across the globe, efforts are ongoing to identify those management practices, particularly in agricultural systems, that can be used to increase soil organic carbon. How can we promote photosynthesis, which pulls CO2 out of the atmosphere and converts it to plant biomass, and then take that plant biomass and promote its deposition in the soil and its decay into its fixation or storage in soil organic carbon? The great thing is that by promoting the rate at which carbon is pulled out of the atmosphere and put into the soil, not only helps us reduce atmospheric CO2 concentrations, but increased soil organic carbon also has other benefits. Soils with higher organic carbon concentration tend to have better soil structure, so water infiltrates better, they're less resistant to erosion. Increasing soil organic carbon is also associated with better water and nutrient retention, higher soil biodiversity, and even better improved crop yields. So increasing soil organic carbon is a real win-win situation. Irrigation is strongly tied to food production in large regions of the world. From over the last 60 years, irrigated agriculture has more than doubled, from about 160 to over 320 million hectares across the globe. Irrigation can help us increase crop yields, and it also expands the area on which we can conduct agriculture, which is really important as we try to feed our growing global population. Canada alone uses 2 billion meters cubed of water for irrigation. Here in the Okanagan Valley, the climate is semi-arid. Without irrigation, the vegetation tends to be quite low productivity. You can see that in the foreground of the photo in the middle of this slide, where there's sagebrush, there's grasses, and the occasional pine across the landscape. Whereas on irrigated agricultural fields, plant productivity is much higher. Because irrigation increases plant productivity in the Okanagan Valley, we were curious to understand how this affected the distribution and the quantity of carbon in the soil. Thanks to irrigation, we are able to grow apples, cherries, and wine grapes, and a host of other crops that the Okanagan Valley has become famous for. Irrigation water is drawn not only from the network of lakes that run approximately north-south along the Okanagan Valley, but also upland reservoirs, which are used to capture spring snowmelt. We wanted to know, as I mentioned, how does the size and distribution of the soil carbon pool vary among irrigated, perennial, horticultural cropping systems in the Okanagan Valley? And we chose apple and cherry orchards and wine grape vineyards as examples of typical growing systems here. To do this work, we collected samples from over 80 sites along the Okanagan Valley, 24 drip irrigated vineyards, 17 drip irrigated apple orchards, 19 micro sprinkler irrigated apple orchards, and 21 micro sprinkler irrigated cherry orchards. And these irrigation methods are pretty typical for what you find in the Okanagan Valley. At each orchard and vineyard, samples were collected from 2 general locations. In the crop row, which is typically maintained weed-free using herbicide, and so it only supports the tree or vine, and the drive row, which is where tractors typically drive. It usually grows drought-tolerant grasses and weeds that were either purposely planted or come up as volunteers. Samples were collected at 3 depths: 0 to 15 centimeters, 15 to 30 centimeters, and 30 to 60 centimeters. And all of the samples were subjected to analysis of total organic carbon and the 13C isotopic composition of the carbon, which I'll come back to. So I'm going to spend a fair bit of time on this slide here because it's basically a summary of most of our results. Across the y-axis are sample points representing the 3 sample depths I mentioned: 0 to 15, 15 to 30, and 30 to 60 centimeter samples. And across the x-axis is the total organic carbon measured in the samples collected in each of these locations. So each panel represents a different cropping system. There's drip irrigated apple orchards and vineyards, and there's micro sprinkler irrigated apple orchards and cherry orchards. There's a couple things I want to draw your attention to. The first is that the concentration of total organic carbon tended to be highest near the soil surface in all cropping systems. And this isn't too surprising. We know that leaves and tree branches and unharvested fruit all falls at the soil surface, and it's there where it's broken up and converted into soil organic carbon. So the concentration of organic carbon tends to decline with depth because in deeper soils, it's only a few roots that typically reach down to 30 or 60 centimeters. It was really interesting to notice the strong differences in organic carbon in samples collected from the crop rows and the drive rows, particularly in the apple orchards, where soils in the drive rows tended to have quite a lot more organic carbon than those in the crop rows. Differences weren't as strong in the vineyards and the cherry orchards, but what was really interesting is that the vineyards tended to have lower organic carbon in soils of either location, whereas the cherry orchards tended to have higher organic carbon concentrations in both the crop and drive rows, particularly at the soil surface. One other interesting point was in the drip irrigated vineyards. You can see at 36 to 60 centimeters depth, it almost appears that the organic carbon concentration is actually higher in the crop row than it is in the drive row. These differences are pretty small, but the trend was strongest in the vineyards, and we think that may be associated with root inputs deeper in the soils of the vineyards. So this is a model that kind of helps us explain why we think we're seeing what we're seeing. In apple and cherry orchards, the tree roots tend to be concentrated near the soil surface. And in the crop row, there are very few organic carbon inputs. The leaves tend to blow away. There are very few inputs from weeds because herbicides are used typically to maintain a bare surface soil to reduce competition between weeds and the crop row. And so the inputs of carbon tend to be quite low. In vineyards, there are deeper inputs from grape roots, which tend to grow and explore more thoroughly the soil volume than apple or cherry roots. And this contrasts with the dry row vegetation, where you see lots of weeds and roots that have abundant root biomass, lots of carbon going into the soil. And because this carbon is these roots are concentrated near the soil surface, carbon inputs are particularly high at the soil surface, and they decrease with soil depth. So we think that the differences that we're seeing in organic carbon between crop and dry rows is strongly driven by the differences in the amount of vegetation growing in these two locations within the orchards and vineyards. To back up our explanation, we decided to use measurements of the 13C isotopic composition. So I'm just going to back up and remind you what 13C is. We take a brief foray into the world of carbon isotopes. Back to grade 11 or 12, you probably remember that carbon has two main isotopes. Most of the carbon in the world is carbon-12. It has 6 protons and 6 neutrons. But about 1% of all carbon is carbon-13. It's slightly heavier because it has an extra neutron. And because of this extra neutron, it's considered a little bit heavy. It's a little slow, and it undergoes biological and chemical reactions just slightly slowly or less often than carbon-12. We call this process natural discrimination. And because of natural discrimination, carbon-13 tends to build up in some types of carbon, and it tends to be depleted in other types of carbon. I'll show you what I mean in a minute. We express the amount of 13C in a carbon-containing compound using the delta notation. The delta notation expresses the abundance of 13C relative to a standard that's called PD belemnite. PD belemnite is a rock deposit. It's basically fossilized squid that were naturally rich in 13C. And because they're naturally rich in 13C, they have a very high 13C composition. Most carbon-containing compounds that we see today are less abundant in 13C than the standard. So this PD belemnite standard has a delta 13C of 0. It's been set to 0. Most carbon-containing compounds have values less than that. So atmospheric CO2 tends to have a delta 13C of about -9-ish, whereas carbon-containing soil pools tend to have even less 13C. Fresh roots and leaves tend to be very depleted in 13C. This is because photosynthesis discriminates against 13C. When photosynthesis occurs and plant biomass is produced, it tends to produce plant biomass that's very depleted in 13C. Fresh plant biomass in the systems that you'd find around here tend to have a 13C isotopic composition of about -28. But as these fresh roots and leaves decay and are converted to soil organic carbon, they become increasingly enriched in 13C. With time, they work toward an isotopic composition of about -22 or -24. So we can use this information to get a better understanding of how fresh the organic carbon is in a soil. If it's very close to -30, that indicates that the soil organic carbon is dominated by really fresh inputs. If the isotopic composition is closer to -20, it indicates that the carbon has been more processed, more decayed, it's less fresh. So these data are the results of the 13C analysis of the soil samples we looked at before. Now, instead of looking at the concentration of total organic carbon in the soils, we're looking at their isotopic composition to get a better understanding of how fresh or microbially processed the carbon is in each cropping system and each location within these cropping systems. So you can see that near the surface, the isotopic composition of organic carbon in the dry rows tends to be relatively depleted. It's about -24, closer to fresh inputs of roots and leaves. In the crop row, the isotopic composition of those surface soils is more enriched. It tends to be about between -23 and -22. This suggests that there's more fresh inputs in the soil surface in dry rows than in the crop rows. But as we sample more deeply, the carbon becomes more enriched, suggesting that the carbon down in these deeper horizons is older and more microbially processed, and therefore more 13C enriched. It's really interesting, especially in the deeper soils from the vineyards, but you see this to some degree in the cherry orchards as well, that the isotopic composition of samples collected in the crop and dry rows tends to reverse in the deeper soils, indicating that there's greater root inputs in the crop rows than in the dry rows at depth. And this is consistent with what we also found in the organic carbon concentration data that we looked at earlier, which suggests that cherry roots and even more so grape roots tend to grow more deeply and deposit organic carbon deeper down under the crop row soils. So the isotopic composition data support our hypotheses or our explanations drawn from the organic carbon concentration data. In the crop row, there's a lot of carbon inputs, mostly from roots and near the soil surface. But grape roots and maybe to some degree cherry roots also grow more deeply and add carbon deeper down in the crop row. Carbon concentrations tend to be higher in the dry row, particularly in apple orchards, because there's a lot of dry row vegetation growing there. The grasses in particular produce a lot of roots that are concentrated near the soil surface, and they add a lot of carbon to those soils. We take these data and scale them up to carbon stock data, which is corrected for the density of the soil and the sample depth. We can calculate the stock of carbon in each kind of cropping system. We also corrected these data for the widths of the dry rows and the crop rows. I should mention that in apple and cherry orchards, about half the land surface is under dry row and about half of it is under crop row, which is slightly different than what we saw in the grapes, where about 60% of the land area is covered by dry row and about 40% by the crop row. Overall, the quantity of carbon, or the carbon stock, tends to be lowest in the vineyards. The overall carbon stock in vineyards is about 48 megagrams of carbon per hectare. This is very different than what we see in the apple orchards and the cherry orchards, which tended to be about 66 and about 70 megagrams of carbon per hectare, respectively. You can see in these data as well that the differences between the crop and the dry row are most strong in the apple orchards than in the vineyards or cherry orchards. So what does this all mean? I think we can conclude from our findings that crop and dry row vegetation, both of which can be facilitated by irrigation, are really strong determinants of how much carbon is in the soil and where it's distributed. Drive row vegetation plays an important role in increasing the amount of carbon bound in those soils. Soil carbon content tends to be greatest in cherry orchards, followed by apple orchards, and then vineyards. So what might this mean for the future if we were really interested in trying to understand how cropping system affected the amount of carbon stored in these soils, especially with the perspective of climate change mitigation and soil quality changes? In 2006, 2007, and in 2015, 2016, land use surveys were conducted across the Okanagan Valley, and they showed that cropping systems in this valley are by no means static. Growers are always changing what they're growing and where. Over this period, the acreage planted to apples fell by almost 1,300 hectares. Over this time period, the acreage planted to cherries and grapes increased significantly. And this has implications for how much carbon is tied up in those soils. Based on the data we found, we wanted to know what would happen if all drive rows were managed like those in apple orchards. We calculated these data using the land use survey data from the 2015-2016 survey. We just did some very simple math to show that if all the drive rows were managed like those in apple orchards, within cherry orchards, we get a very small percent change, because cherry systems also tended to have quite high carbon in their drive rows. If the grape vineyards were managed, if the drive rows in vineyards were managed like those in apple orchards, which tended to have the highest amount of carbon, we would have really strong increase in the amount of carbon stored in those soils, almost 23%. We're also curious about another scenario. What if we took all of the orchards and vineyards included in this study that fell under these categories in this study and converted them to a single crop? So if we converted all of the vineyards and cherry orchards to apple orchards, we would get about 13% loss in carbon, because cherry orchards overall tended to have the most carbon. They had high carbon both in the drive row and the crop row. If we converted all the apple orchards and grape vineyards that fall within the categories included in this study into cherry orchards, we get almost 17% increase in carbon storage, because cherries tended to have the greatest amount of soil carbon overall. But if everything was converted to wine grapes, we get a strong reduction in the amount of carbon stored in these soils. Finally, we were curious about our options for promoting soil carbon storage in these horticultural systems. If we decided these systems are great for growing crops, and maybe simultaneously we can promote carbon storage, both for climate change mitigation and improving soil quality. There are many options, and I've just listed a few here. We could plant cover crops that promote soil carbon storage and at the same time require less irrigation water. In particular, we could consider deep-rooted cover crops, which really pump carbon deeper into the soil. And this is work that colleagues at AAFC are already looking at, and it's also research that is being conducted in many other countries, particularly New Zealand. What about in the crop row? What could we change? We could apply organic amendments like organic mulches or compost, and research on these kind of management practices is being conducted by Agriculture and Agri-Food Canada. We could use organic mulches to suppress weeds instead of always relying on herbicides, or maybe we could reduce the width of the herbicide strip. Maybe we could investigate pruning systems or rootstocks that can better tolerate competition from surrounding vegetation, so we wouldn't have to worry so much about maintaining the crop row weed-free. What are some next steps that we're looking at with this research? First, we'd like to assess the persistence of the carbon that we've added to these crop and drive rows. It's really important to know whether that higher amount of carbon in the drive row will stick around. if it isn't persistent and we have to maintain management practices to keep that carbon where it is, we really need to know. We'd also like to know what the saturation deficit of these soils is. How much extra carbon can we add to them? Or how do they become saturated at the soil surface? And finally, a lot of the orchards and vineyards that we studied are irrigated using irrigation water drawn from the lake system. And this irrigation water has bicarbonates in it, inorganic carbon, which tends to form crusts on things like the lime scale in your kettle. And we're curious whether this inorganic carbon forms crusts around the organic carbon that's deposited in the soil, making it less susceptible to decay. But it's really important to remember that increasing soil carbon is only one possible tool for mitigating climate change. Soils only have a finite capacity to retain soil carbon, and we have to continue to maintain good soil management practices to protect soil quality and to keep the soil carbon that's already there. We need to exploit that win-win and help us understand how increasing soil carbon might help us reduce the amount of other inputs like fertilizers that we need to use. And finally, I really want to thank the very many Okanagan Valley orchardists and vineyards who let us sample their soils. There were so many of you. You were so curious and interested in our research, and we're really grateful that you let us come on site and ask you so many questions and collect soil samples from you. The work also would not have been possible without many, many summer students and technicians who helped us along the way from both UBC Okanagan and Agriculture and Agri-Food Canada. Thanks. I hope you found the talk interesting.